These are the many phases of me.
I'm interphase. When the cell prepares for division, interphase has three distinct phases. The G1 phase, called the first gap stage. The cell cycle grows and produces new organelles and other structures needed for DNA replication. Replication of the centrioles to produce two centriole pairs. The S phase, called synthesis. The 46 double helix strands of DNA are replicated. Forming DNA requires large numbers of deoxyribonucleotides in the enzyme DNA polymerase. They are located in the nucleoplasm. The G2 phase, called the second gap stage. During this phase, centriole replication is completed and enzymes and other structures needed for cell division are synthesized. This is interphase.
I am prophase. Chromosomes appear due to coiling of chromatin, nucleolus breaks down, spindle fibers begin to form from centrioles. Centrioles move towards cell poles, and the nuclear envelope breaks down at the end of this stage. This is prophase.
I'm metaphase. Single fibers attached to the centromeres of the chromosomes extending from the centrioles. Chromosomes are aligned at the equatorial plate of a cell by spindle fibers. This is metaphase.
I am anaphase, arguably the coolest phase. Sister chromatids are separated by spindle fibers and move toward opposite ends of the cell. During this process, centromeres that held sister chromatids together separate. Each sister chromatid is now a chromosome with its own centromere, and cytokinesis begins. This is anaphase.
I'm telophase. Chromosomes uncoil to form chromatin. A nucleolus reforms within each nucleus. Spindle fibers break up and disappear. New nuclear envelopes form around each set of chromosomes, and cytokinesis continues as cleavage furrows deepens. This is telophase.
I'm cytokinesis. This is the division of the cytoplasm between the two newly formed cells. This phase usually begins early and overlaps with the anaphase and telophase of mitosis. A ring of the microfilament proteins on the inner surface of the L's plasma membrane contracts at the cell's middle. It pinches the mother cell into two separate cells in a manner analogous to the tightening of a belt. The resulting cleavage furrow that appears indicates where the cytoplasm is dividing. Two new daughter cells are formed and cell division is complete.
We're finally done! Hi, this is Lainey from NSU News, Channel 16, here to investigate the death of Marcella Smith and to rule it as a programmed death or a homicide.
Well, what would rule this death as a programmed one?
This programmed cell death is called apoptosis and has very distinct steps. The destruction of DNA polymerase to prevent the synthesis of new DNA, digestion of the DNA into small fragments, digestion of the cytoskeleton, thus destroying structural support for organelles and the nucleus. The cell appears to shrink and becomes rounded and the nuclear shape changes. Condensation of the cytosol and the destruction of organelles, specifically the mitochondria, to deprive the cell of ATP needed for energy requiring processes. The formation of small irregular bubbles on the plasma membrane surface, and the initiation of other plasma membrane signals to stimulate destruction of the cell externally by phagocytic cells.
Oh, so this occurs to promote proper development and to remove harmful cells, and it causes no inflammation and no secondary tissue damage.
Yep.
Well then, what would make this death a homicide?
Well, the cell would have been killed by harmful agents or mechanical damage. It can be through infection, poisoning, overheating, or lack of oxygen. The cell can swell up and its contents can leak away. This can cause damage to the surrounding cells.
Well, this sounds like it could get messy. We better get out of here.
Well, let's stick around and see if we can find some eyewitnesses to talk to. This person looks suspicious. Let's go talk to them. Hi there. Do you have any information regarding what might have happened here last night?
No.
Where were you last night between 11 p.m. and 2 a.m.?
Well, I was at home in my cellar.
Can anybody verify that?
No, I was all by myself. Why are you asking all of these questions? Wasn't she aging anyways?
Well, how do you know that?
Well, she had reduced metabolic functions and a reduced ability to maintain homeostasis. She was no longer functioning properly, and she had changes in her structure of chromatin and chromosomes.
Interesting. Okay, never mind, you're fine to go. Evidence has been found that leads us to believe that this was a programmed death. Looks like our work here is done.